Hello, welcome to this video, which is a little bit of a focused look at the microbiology of the GI tract. It's not really intended to be masses and masses of detail, but just a little bit of an overview to support some of the anatomy and physiology teaching you've got at the same time. We're going to go through a few things, and when learning about the GI tract, it is important to keep microorganisms in the back of your mind, giving their kind of given that what we know about their impact in your everyday health and also in disease as well. We'll go through a few things with the really important things I've highlighted in yellow. So simply, you should be able to say where we find the highest and lowest numbers within the GI tract. That's true both of bacteria, viruses, and other things as well. You should be able to define adhesin and biofilm. You should, as a bit of revision potentially, you should be able to define what we mean by gram-positive, gram-negative, and anaerobe, and we'll relate that back a little bit to the diversity of things that grow inside the GI tract as well. We'll go through a little bit about oral microbiology. So when we're thinking about the GI tract, naturally the emphasis tends to be a bit more on the small and large intestine, but actually the GI tract starts with the mouth, and oral microbiology has big impacts elsewhere than within the body. And I'll give one specific example of a link between oral microbes and other infections, which is quite surprising. And then lastly, I'll just take you through a little bit of detail with Helicobacter pylori. So this is a specialized bacterium which grows within the stomach. And we'll use H. pylori as an example to highlight a couple of things, or a couple of features which are found within, let's say, good pathogens. More on that a bit later. We'll start by thinking about where do we find these things. Your mouth is teeming with life. Huge numbers of bacteria grow there. We see different types of bacteria, gram-positive, negative, anaerobe, facultative anaerobe. The esophagus and stomach, you see relatively low numbers. They're not sterile. The esophagus is low, food is transiting through, so you don't really get much growth happening there. And you also get turnover of cells, so cells slough off, and when they do, they take any bacteria that are stuck to the surface, they take them away. Then they come to the stomach, and the stomach obviously has low pH, and unsurprisingly that kills off large numbers of cells. Now, it's not completely sterile, so very small numbers of cells will survive, and they will make it through to the small intestine. And as mentioned, there's one specific pathogen to know of, that's Helicobacter pylori, which grows within the stomach. So it's a very specialized pathogen in that sense. There are a few others which are a bit more acid tolerant. And when they emerge through to the small intestine where the pH comes back up and is neutralized, they're in a slightly higher number. So a good example is lactobacilli. These are bacteria which grow within the small intestine, which can survive the stomach acid a bit better. You'll see on the graph that the line with the intestine kind of slopes up, and you can kind of think of the small and large intestine as a bit like a conveyor belt. So material is moving along. As it's moving along, it's digesting, and then cells are growing on that material, and those numbers are creeping up. And by the time you get to the large intestine, it's packed full of life. What types of microorganisms do we find there? So there are three main types within the GI tract to note. So we have bacteria, viruses, and yeasts. We don't really tend to find parasites growing normally within the GI tract. So the presence of worms, for example, would not be a normal finding. Just on the screen here, I've just included a little bit more detail. So we'll have different types of bacteria, so gram-positives, gram-negatives. You may hear sometimes the word enteric organisms, and anaerobes as well. Viruses are mostly bacteriophages. They are viruses which prey on the bacteria. And then the yeasts, these tend to be, there's less of a priority to think about for now, but they are cells which are very good at breaking down plant material, and some evidence that they have quite potent anti-inflammatory effects, i.e. yeast cells will prompt an anti-inflammatory response from nearby immune cells. All three are normal findings within the GI tract. So the presence of viruses or the presence of yeast shouldn't become as any surprise, that they're normal. And in some cases, there are some hypotheses which say that certain diseases, certain inflammatory bowel disorders come about when the balance of these three communities goes wrong, and you get what's called a dysbiosis, and you get potentially an inflammatory reaction, which is then difficult to control. So when we're thinking about GI tract, and again, it's a good chance to take a step back and revisit the main classes of bacteria. So thinking about antibiotics and where we find different organisms within the body, there's one class here which isn't really relevant to the GI tract, and that's the atypicals. So the atypical bacteria, which are maybe unusual cell walls or unusual life cycles, they're more restricted to genitourinary and respiratory tract. So we can actually get rid of them. But the other three are things to consider. And I've mentioned them here. So anaerobes, you'll have dedicated anaerobes, which live within the oral cavity, those which are found more or almost exclusively within the GI tract. In terms of gram-positives, there are some which are unlikely to be found in the GI tract. So staph and strep, it's unlikely. It's not impossible because they do sometimes escape in through the skin, through the mouth. But you may see this organism Enterococcus. Enterococcus is a cousin of Streptococcus. In fact, for a long time, it was also called Streptococcus. And with the gram-negatives, it's these at the top which are important to note. So enteric gram-negative rods, such as E. coli. And some of these other things are much less important for the GI tract. So you wouldn't normally expect to find some of these respiratory pathogens and organisms, Pseudomonas, you wouldn't expect to find within the GI tract. Or if you did, that would be a very unusual finding, and you'd probably want to think about what's going on there. So again, good chance to revisit the fact that we have different classes of microbes and that they can be found in different sites within the body. Let's start with the mouth. Oral microbiology is a dedicated area. You'll actually have some teaching on that in year 2 by one of the colleagues from the dental school. And I've just included this figure from a paper which came out last year just to show and highlight the fact that oral microbiology and specifically oral bacteria have massive systemic effects within the body. So evidence that they have impacts with Alzheimer's disease, with cardiovascular disease, certain types of esophageal cancer, for example, and evidence that they have, unsurprisingly, they cause infections and inflammatory disorders within the mouth as well. So when we're thinking about oral microbiology, it's not just about tooth decay and stuff for dentists to consider, but you should also be thinking about and noting the fact that, of course, the mouth is connected to the rest of the body, and so it makes sense that these large numbers of cells can potentially enter the bloodstream and then move around inside the body, but also that their pathogen-associated molecular patterns or molecular factors can also spread and can potentially trigger inflammatory disorders and reactions within the body as well. So the figure that I've got here, just as a bit of reassurance, not necessarily anything to remember or take away, it's just to note that the things which live inside the mouth have impacts elsewhere in the body. Probably one of the most important and widespread infections and pathologies which is seen worldwide is tooth decay. And tooth decay can start out a little bit, let's say, superficial. So you may get kind of caries or pitting, so where the surface of the tooth becomes damaged, but can then quickly escalate and become much worse if left untreated. Caries is one of the most important infections just to be mindful of. Again, you're not expected to be an expert in caries because that's why we have dentists. But essentially what happens is you get bacteria colonize the surface of the tooth, and they can also colonize along the gum line as well. They come into direct contact with tissue. So these bacteria, what they begin to do is they begin to ferment the food that you eat. They're converting that food as part of their natural respiration into acid, which can begin to damage the surface of the tooth. Now, normally what happens is that when you're doing things like brushing your teeth and you're using fluoride, that helps to remineralize the surface of the tooth. So you can, in some way, correct some of that damage by normal oral hygiene. But what can occur is that if this is left unchecked, if you don't see the dentist ever, if you don't do these things, then what you can lead to, just to go back, is you can actually start to see some erosion much further down. Before we go any further, let's quickly think a little bit about how bacteria bind and adhere to surfaces such as tooth. This is an image which we cover in year 1, so just the kind of generic features and generic cell structure of bacteria. And when we're thinking about how do bacteria stick to surfaces, the one thing which people always highlight in these images, of course, is the pili. And pili are important at allowing bacteria to adhere to surfaces, but there's something else that's missing. And they are a series of proteins and structures found over the surface of the cell. And these go by the kind of generic umbrella term of adhesins. These are, as the name sort of implies, adhesive molecules found over the surface of bacteria. And they tend to be more cell surface or cell type specific, allowing bacteria to stick to and colonize particular types of cells or particular surfaces within the body. So, for example, oral bacteria which can colonize the tooth and can colonize the gum do so because they can bind to and stick to those surfaces in the first place. It's quite complicated, and I've just shown this image just to show you a little bit of the complexity of what we're seeing. But essentially, adhesins are a big range of proteins, and in some cases other glycoproteins or sugars that stick off the surface of the cell that are cell dependent, so bacterial cell dependent, which look for host cell specific or host surface specific molecules in return. There's not a lot to say beyond that, other than to kind of think a little bit about it as, imagine Velcro in your mind. So you've got a surface of Velcro over the surface of the bacterial cell that allows it to attach to other host cell receptors. Go back to this image. So what can happen is that in time, when you get colonization of bacteria over the surface, you can see inflammation. And this is what's called gingivitis. So sometimes if you see a little bit of blood when you brush your teeth, this can result in what it can be a sign of gingivitis. So you've got an inflammatory reaction, so you've got increased blood flow to the presence of all these microbial antigens over the surface of the tooth. But what can happen is that over time, the bacteria can move down and can colonize much further, deeper into this pocket, and you can get what's called periodontitis, or perio, which is referred to as dentists, is also very common, but is more serious because it can lead to tooth loss. And when you see lots of tooth loss, what you're losing in effect is one of the major innate barriers to infection. The tooth, just looking at it here, effectively is a plug stopping all those large numbers of oral cells from entering the body. When they disappear, it allows more bacteria into the body, and that's when they can begin to circulate and colonize and can potentially have some pathological effects elsewhere. Let's stop for a second. So oral bacteria then, we've already covered adhesins. Let's think a little bit about biofilms. So oral bacteria are very, very good at making these things. What a biofilm is in effect is an extracellular layer of polysaccharide. You can kind of think about it as a layer of slime. It's made of polysaccharide, it's made of protein. Sometimes you'll find DNA and fat in there as well. And what it allows and what it does is it essentially protects the underlying cell from environmental stresses. So it physically protects them against mechanical forces, so fluid, air, but it also protects them against antibiotics and other pharmaceuticals. And a couple of important examples are these organisms. So one is called Streptococcus mutans. This is also referred to more generally as viridans strep. By itself, not necessarily a particularly nasty pathogen, but can cause some quite low-grade infections elsewhere in the body. And then also you get some skin flora can do this as well, or can produce biofilms, so such as things like Staphylococcus epidermidis. So you've got bacteria that can stick to surfaces and produce a biofilm. Let's think about this in a bit more in a surprising way. So all bacteria can cause infections of implanted devices and can cause infections of other sites within the body. So what you have is your bacteria. So in this case, a good example is strep. So Streptococcus colonizes the surface of the tooth, where it will do what it's good at, which is make a biofilm. These bacteria can enter the bloodstream. They can do so by something as innocuous as brushing your teeth. So you graze the gum line, the bacteria can enter, and they can then begin to circulate. And when they circulate, they can potentially, in some cases, find surfaces that they can land on. Now, if you don't have a device implanted, the bacteria will circulate around and will probably be cleared out by the immune system. But in some cases, implanted devices, that could be like a stent, as I've shown here, or it could be a joint replacement, or sometimes scar tissue that's laid down internally as well, can be colonized by these things. And the bacteria land on the surface and they do what they're good at, which is that they make essentially dental plaque, only it's not being made on the tooth, it's now being made on the surface of a heart valve or a stent that's been implanted. And that's difficult to treat. It's difficult for your immune system to clear, and it's also difficult to treat with antibiotics. It's a good example of how all bacteria connect elsewhere in the body. And this is something and an idea which we'll revisit a bit more in year 2, but I just kind of want you to think about this if you're watching this in year 1, that these organisms have a role. Again, it's a good example of where an organism can be, that's found in one part of the body, can be a potentially low grade or fairly innocuous organism at one part, but can cause some quite serious disease at another part of the body. Back to this image. We'll finish, and the last part of this video considers the stomach, so relatively low numbers of cells. And a good stomach specific pathogen to consider is Helicobacter pylori. A very large number of people actually have this, and it lives asymptomatically inside of you. You may not know that you have it. It's acquired through environment, water, food. Most of the time you're asymptomatic, but if you have it, you have the potential of developing an ulcer. What is Helicobacter? It's a very specialized human pathogen that has a number of different features amongst it which allow it to be very good at colonizing and living within inside your stomach. So the first thing that it has is lots of flagella. So the tails, which you can see here, this allow it to propel itself very, very quickly. So when it enters the stomach and when it's swimming around, it can move very, very fast over to the lining of the stomach. Remember that you have a barrier which stops the stomach acid from digesting the rest of your body away. And that barrier is a kind of like a nice convenient home that these cells can live within. It also has adhesive molecules. So again, it stays there because it allows itself to stick to the surface of epithelial cells within that. But not only that, it's got a series of what are called virulence factors. Now, this is a term which you may have not come across yet. Essentially, this is another broad term which refers to the molecules, proteins, enzymes which facilitate disease, i.e. facilitate virulence. So these, in this case, are proteins which are injected into nearby cells, which actually trick epithelial cells into releasing cytokines and inducing an inflammatory reaction. It seems a little bit counterintuitive. Why would a bacterium provoke an inflammatory reaction? Because surely that would result in lots of, white blood cells coming to the area. Well, it does that because increased blood flow is also good for the organism. It means more food, it means sometimes more iron, it means more oxygen potentially, and that can all be good news for for the bacterium. And lastly, another feature which is also important diagnostically in this thing is that it produces an enzyme called urease. And the urease helps to neutralize the stomach acid by producing ammonia, and it also produces carbon dioxide in doing so. The ammonia neutralizes the stomach acid. It forms a small bubble around the bacteria, which is relatively neutral, which again allows it to survive. But the CO2 that's produced can be detected, and there are various breath tests you can use. So rather than using an endoscopy, you can use a breath test looking for the CO2 released specifically from the bacteria. And how we do that is using a special type of radio-labeled carbon, and that's a conversation for another day. But a few specific features allowing it that have allowed it to grow and live as a specialist pathogen within the stomach. So just as a quick summary of what's going on, the bacteria can detect and swim quickly over to the lining of the stomach. Once it's there, and along the way, it can produce the urease and other toxins to help protect itself, injecting nearby cells to induce inflammation, and then also in doing so, it can increase cell turnover, which leads potentially to an ulcer, but also can lead to certain types of stomach cancer. And it's an example of a cancer which can be treated with an antibiotic, because once you've killed the bacterium that's provoking that inflammatory action, actually the immune system can move in and clear and can resolve the lesion that's formed. So that was an introductory perspective. A few key things to say: where do we find the highest and lowest numbers? A few words to define. Some of them might be new, but there's a few old ones which might be a bit more of a useful revision topic. Everything else we revisit at other points within the course, but just a few introductory ideas as well. Any questions, get in touch. The next video will consider the lower parts of the GI tract, so the small and large intestine. Thanks a lot.